I am Louis Gruber, a founder, CEO, and CSO of SIWA Therapeutics, Inc. I'm pleased to be here today to tell you about SIWA's comprehensive treatment for aging and aging-related diseases through removal of all types of oxidatively damaged dysfunctional cells, including but not limited to senescent cells and cancer cells. We have a therapy we believe to be applicable to virtually all aging-related diseases, including cancer, not only without side effects, but also with the ability to reverse aging-related damage by promoting regeneration. In the purview of metabesity, as the constellation of interconnected diseases with metabolic roots in an obesogenic context, a common thread among aging-related diseases treatable by our monoclonal antibody, SIWA 318H, or 318H for short, is an underlying metabolic disarrangement of aerobic glycolysis accompanied by oxidative stress. By binding 318H selectively targets a carboxymethylysine epitope on dysfunctional disease-causing cells having a combination of an abnormally high level of glycolysis accompanied by oxidative stress. Carboxymethylysine was chosen as the marker targeted by 318H because it is an accepted biomarker for oxidative stress and oxidative damage, a root cause of aging. It appears on dysfunctional cells, including cancer cells, senescent cells, and microbially-infected cells, all of which shift from predominantly oxidative phosphorylation to glycolysis. This process is the source of glyoxyl, the precursor of carboxymethylysine, that is modified by oxidative stress to become the biomarker for 318H. As expected, 318H has been shown to bind to all types of cancer and senescent cells tested, including renal tubular epithelial cells made senescent, as well as Alzheimer's and Parkinson's disease cells. And last year, we showed 318H bound to cells infected by SARS-CoV-2, the virus giving rise to COVID-19, and also to cells infected by Influenza A virus. Reports indicate that targeting senescent cells alleviates obesity-related metabolic dysfunction, and that inguinal adipose tissue in obesity elicits a high level of insulin resistance, a factor in type 2 diabetes. As noted later in this presentation, 318H has been used to successfully target senescent cells in inguinal adipose tissue, which is consistent with the externally reported increased formation and accumulation of carboxymethylysine in inguinal adipose tissue in obesity. By way of background, we founded SIWA to develop therapies for slowing and reversing the aging process, specifically by removing dysfunctional cells like cancer and senescent cells, which are involved in aging-related and degenerative diseases. We developed 318H, our proprietary monoclonal antibody, by focusing initially on a mechanism that enhances immune surveillance and the destruction of senescent cells by targeting them with an antibody for immune destruction. Given the role of cumulative oxidative damage in the aging process, I identified an advanced glycation end product marker for oxidative stress that was found on both cancer and senescent cells. Our marker is an oxidative side product of glycolysis, and both cancer cells and senescent cells conduct glycolysis to an elevated degree, known as the Warburg effect. Related to increased infectious disease morbidity and mortality among the elderly, We also know that virally infected cells have the same elevated glycolysis and oxidative stress. This combination of effects produces our biomarker. Last year, SIWA was named as a key player in anti-aging drugs by the MIT Technology Review in its list of the 10 breakthrough technologies of 2020. 318H targets cells with elevated oxidative stress and elevated aerobic glycolysis. As an indication of 318H's broad effect on health span, oxidative stress and aerobic glycolysis correlate to the leading causes of death for the elderly, as identified by the CDC, and all of the common chronic conditions of the elderly listed by the Society of Consultant Pharmacists. Also, in addition to our confirmation that the 318H biomarker is found in association with P16INK4A, a well-known senescence marker, it has also been reported to be found in association with other standard biomarkers of cellular senescence. When we looked at diseases to pursue commercially, we set a few parameters, the most important of which was that it be a life-threatening disease with unmet medical need, that is, that it be eligible for FDA fast-tracking. We have focused as a company and with a major institute on aggressive cancers. By clearing the tumor microenvironment of both cancer cells and the senescent cells that support them, we believe 318H can be a therapy for treating virtually all cancers and for stopping cancer progression, metastasis, and relapse, not only without side effects, but with the ability to reverse side effects of other cancer therapies with which 318H may be co-administered without interference. At the end of September, we reported on our successful in vivo PDX pancreatic cancer study with 318H in humanized mice having patient-derived explants. We reported on that at the American Association for Cancer Research Pancreatic Cancer Conference. I will talk about those results on a later slide. We are planning for a glioblastoma study at the same institute. By clearing the tumor microenvironment of both cancer cells and senescent cells that support them, we believe 318H can be a therapy applicable to virtually all cancers for stopping cancer, progression, metastasis, and relapse, not only without side effects, but with the ability to reverse the side effects of other cancer therapies with which our product may be co-administered without interference. We expect to file an investigative new drug application by the third quarter of 2022, and we expect that a phase 1 basket trial in solid tumors, which would be done with the Institute, will begin shortly thereafter. Charles River Laboratories has completed for us an initial tolerability study in non-human primates in preparation for filing an IND application with the FDA. No adverse effects were observed. We have also completed histological and pharmacokinetic studies. And we have begun the process of moving from small batch production of 318H to a product satisfying current good manufacturing practices of the FDA's formal regulations. We are actively seeking licensees or partners to help us expedite this development process. This slide shows images of some of the binding results of our monoclonal antibody with cells from tissues associated with aging-related diseases. We have observed binding of our monoclonal antibody to pancreatic cancer cells, glioblastoma cells, Alzheimer's vascular microglia, and human osteoarthritic chondrocytes. Among other results observed but not shown is the binding of 318H to alpha-synuclein-associated cells from patients afflicted with Parkinson's disease. We first showed in vivo in mice that a 318H mouse homolog both targeted and removed senescent cells and promoted muscle regeneration. This slide shows the result of injecting a mouse version of 318H into very old, naturally aged mice over 20 months old, an age at which untreated mice exhibited substantial muscle wasting as measured by the mass of their gastrocnemius muscles. In the slide, the results are showed alongside comparisons with untreated old mice and with young mice a few months old. Our mouse homolog was administered twice daily, once a week for 3 weeks, and it reduced senescent cells to the level of young mouse controls, as seen in the graph on the left. This reduction in senescent cells was measured in the inguinal adipose tissue of the mice. Consistent with the reported action of senescent cells inhibiting adult stem cells to prevent them from regenerating tissue, a reduction in senescent cells removed inhibition of the stem cells in the mice and allowed muscle mass to be restored to the level of young mouse controls, as seen in the graph on the right. No adverse effects or regressions were observed during a 9-week post-treatment, treatment-free period. The 318H mouse homolog statistically significantly reduced metastasis in another mouse model, a 4T1 triple-negative breast cancer mouse model. It did this consistently with tumor reduction. So that I don't run out of time, I will skip slide 9, slide 10, and slide 11. As an initial study, we had Charles River Laboratories do a non-cGMP pharmacokinetics and tolerability study in non-human primates. The study employed a single intravenous injection of 318H at 3 dose levels, 10 milligrams per kilogram, 30 milligrams per kilogram, and 100 milligrams per kilogram in naive cynomolgus monkeys, ranging in age from 3 to 18 years old, and including males and females. At 10 milligrams per kilogram, a dose successfully used in mouse studies, the half-life of 318H was determined to be 115 hours, or over 4 days. In all of the primates, the 318H was well tolerated with no adverse effects, which is consistent with the lack of adverse effects in all of our in vivo studies. While conducting our study on pancreatic cancer, we observed what we believed was a rather remarkable and very interesting response in one of 3 animals that were implanted with a particular patient-derived explant tumor. One animal had a large tumor and was exhibiting drastic weight loss suggestive of cachexia. The animal was nearing a weight loss close to 20%, which would have required it to be euthanized according to the protocol. I asked if it would be possible to give that mouse, designated number 9, with an injection of 100 micrograms, that is 5 milligrams per kilogram, of SIWA 318H. As this was a dose that had been deemed safe in our toxicology and PK studies in non-human primates, the decision was made to perform a pilot study of efficacy on the treatment of that last mouse, number 9. Interestingly, as a result of this treatment, the mouse had a remarkable recovery of the weight almost to normal weight. Again, although this is an anecdotal event, we believe it is quite encouraging support for our larger scale studies which we conducted in the humanized mouse model. Our initial goal was to develop therapies for slowing and reversing the aging process specifically by removing dysfunctional cells like senescent cells, which are involved with aging-related and degenerative diseases, including cancers. We developed 318H, our proprietary humanized monoclonal antibody, by focusing initially on a mechanism that enhances immune selection and destruction of senescent cells by targeting them with our 318H antibody. 318H is a cytotoxic antibody or immunotherapeutic that attacks a SIWA-identified marker on oxidatively damaged cells. These include not only senescent cells, but virtually all cancer cells and virally infected cells, including those infected by Influenza A and those infected by SARS-CoV-2, the COVID-19 virus. All of these cells have abnormally high levels of aerobic glycolysis, known as the Warburg effect. Our marker is an oxidative side product of aerobic glycolysis. While we continue testing 318H as a cancer therapeutic, we are looking for a 318H infectious disease licensee or collaboration partner to develop 318H as a broad-spectrum antiviral, initially against SARS-CoV-2 and Influenza A infection, which have been observed to occur together and which may provide a combined threat of infection in the flu season. Infected cells and dysfunctional cells associated with cytokine storms are also targeted by 318H. This slide shows confirmation that the 318H mechanism of action is antibody-dependent cellular cytotoxicity, or ADCC. We used a commercially available test with 318H and PSN1 pancreatic cancer cells. PSN1 cancer cells made senescent compared to an inactive isotype control antibody. In summary, by targeting our biomarker, 318H can remove by ADCC not only cancer cells but also senescent cells, virus-infected cells, including oncogenic virus-infected cells and latent virus infections, and also pre-malignant cells. Senescent cells feed the primary tumor, and senescent stromal cells create an immune-suppressive microenvironment protecting the tumor. Senescent cells are also a cause of adverse side effects from other therapies, for example, by cancer or other cells made senescent by genotoxic cancer therapy. SIWA 318H also targets the cancer cells that escape senescence and cause cancer relapse, resulting in an even more aggressive cancer. 318H does not interfere with intracellular processes, for example, the senescence and aerobic glycolysis processes. Thus, 318H can be co-administered with CDK4/6 inhibitors that promote senescence or immunochemo and radiotherapies, as well as having the capability of being a primary treatment by its combination of cancer and senescent cell clearance. In an examination of progression in our pancreatic cancer humanized mouse model, it was observed that both high and low doses of SIWA 318H significantly reduced tumor growth by comparison with administration of the isotype control. Most importantly, SIWA 318H was also observed to significantly increase survival in the pancreatic cancer model. In fact, SIWA 318H promoted complete remission of the pancreatic cancer tumors in the humanized mouse model. Our other key personnel include Misty Gruber, our CFO and CAO and a founder, as well as Dr. Gabriella Rossi, whose work has included clinical trials development and immunological monitoring of patients in clinical trials, as well as process development for the manufacture of cGMP biological materials for early and late clinical trials development. Our contact information is listed here. Thank you for listening today.